Hello students, welcome to EPG Pathshala. I am Dr. Vishwanath Tiwari from Central University of Rajasthan. Today I am going to give you talk about mechanism and regulation of electron transport system that has been taken from paper Thermodynamics of Living System and Bioenergetics. This module is designed to understand mechanism of electron transport system as well as ATP synthesis in mitochondria. We will also discuss how this ATP synthesis in the mitochondria is regulated. So, we will discuss the details of reduction potential, electrochemical gradient, proton motive force and role of different inhibitors and uncouplers in the ATP synthesis. So, in view of this module is divided into following sections. The first one is reduction potential and Gibbs free energy changes for the electron transport. This is important because when we are studying the mechanism of electron transport, we should know how the reduction potential and Gibbs free energy change plays for the electron transport in the electron transport system. The second one is electron transfer in the electron transport chain. So, how the different carriers are participating in the electron transport system. The third one is electrochemical gradient, how electrochemical gradient is created in the periplasmic space, how it is responsible for ATP synthesis. Then we will discuss about chemiosmotic theory, which is given to explain the mechanism of ATP synthesis by ATP synthase. We will also discuss oxidative phosphorylation and P by O ratio. And in the end, we will discuss how inhibitor is works on different complexes of ATP synthase and how inhibitor is different from uncouplers. So, electron transport chain in mitochondrial inner membrane consists of mobile as well as non-mobile electron carrier. Mobile, which can move across the mitochondrial inner membrane, non-mobile, which is fixed in the mitochondrial inner membrane. These two together participate in the electron transfer in the mitochondrial inner membrane through energy transducing membrane, or you can say mitochondrial inner membrane. The same mechanism is also applied when we are talking about the bacterial membrane. So, the mechanism of ATP synthesis in mitochondrial inner membrane and bacterial membrane are very similar. So, the electron transport system is based on standard redox potential. So, before going into the detailed mechanism of electron transport system, it is important to know what is reduction potential. The reduction potential is the capacity of a molecule to hold the electron with itself. That means if a molecule is having low reduction potential, it shows that they have low tendency to hold the electron. That means they have more tendency to donate the electron. Similarly, if a molecule have high redox potential, it shows it have high tendency to hold the electron with itself. That means they have less tendency to donate the electron. That means electron always moves from lower redox potential to high redox potential. In the normal life, you can compare it with the movement of money that is always taking from benevolent to miser. Here electron transfer always taking place from lower redox potential molecule to high redox potential molecule. So, we know that we have an equation that is known as Nernst equation, and Nernst equation is relating Gibbs free energy change with the standard redox potential change, and the equation you know that it is the delta G is equal to minus N F delta E, where delta G represents Gibbs free energy change, N represents number of electron transfer, F is a Faraday constant, and E is standard reduction potential. If you see this equation, you can correlate that there is an inverse relation between Gibbs free energy and standard reduction potential. And you know that electron transfer takes place from lower reduction potential to higher reduction potential. That means when we are talking about electron transfer based on the Gibbs free energy, then it is inversely related. So, electron transfer takes place from higher delta G to lower delta G, or a molecule is having higher Gibbs free energy to the molecule which is having lower Gibbs free energy. As you know that reduction potential and Gibbs free energy are associated with each other, so we can calculate the difference in the reduction potential between a pair of electron donor and acceptor by this equation, where overall difference is equals to reduction potential of electron acceptor minus reduction potential of electron donor. The important point in this equation, you have to remember that acceptor comes before donor. So, A comes before D, so acceptor comes before donor. So, in this way you can remember this equation. So, we are assuming that we have two half reaction. In one reaction, NAD is reduced into NADH with the reduction potential of minus 0.32 volts. FAD is reduced into FADH2 with reduction potential of minus 0.22 volt. In the important point you have to remember here is both are the reduction potential given for the equation where reduction is taking place. But in the question they are asking that we have an equation where NADH get oxidized and FAD get reduced. So, electron acceptor in the question asked is FAD and NADH is electron donor. So, overall standard reduction potential can be calculated using the above formula, but you have to make sure that you are writing reduction potential of electron acceptor, but you have to write the oxidation potential of NADH, which can easily achieved by reverting the sign of reduction potential. That means delta E is equal to standard reduction potential of acceptor minus standard reduction potential of donor, or another word you can say that if you are converting reduction potential into oxidation potential, then you are not going to see the differences, you are going to add both. So, either you can directly calculate difference in the reduction potential using the second equation where delta E is equal to minus 0.22 plus oxidation potential of NADH, or you can calculate by using both the reduction potential where delta E is equal to minus 0.22, which is reduction potential of FAD minus reduction potential of your NADH. So, in this way we can calculate delta E difference, and that delta E difference can be converted into delta G difference by using Nernst equation, by putting number of electron which is transferred here, N which is 2, and Faraday constant which is having value of 96.485 kilojoule. Electron transfer in the complex first will be initiated with the electron donated by the NADH. As you know that electron transfer always takes place from lower redox potential to high redox potential, that means it is clear that NADH must have lowest redox potential in all the electron carrier which is present. Hence, NADH is having reduction potential of minus 0.32, which donate its electron to the complex first and which finally reach to the ubiquinone, which is having standard redox potential of 0.11 volt. You can see here that the overall difference is about 0.43 volts, and this delta E difference is important, and delta E difference is converted into delta G difference, and that delta G difference is responsible for pumping the proton from matrix to intermembrane space, because pumping a proton is not a joke. If you need to pump proton from matrix to intermembrane space, you have to spend energy, and that is coming from the delta G, which is created because of the delta E difference in the different complexes. The one more point that is important to note here is that NADH giving its electron to the FMN and any other electron carrier in the complex first. So, electron always increasing in order of their gradual increase in the reduction potential. Similarly, in the complex second, succinate is an electron donor. Sometime in most of the textbook or in research paper, FAD are also mentioned as electron donor of the complex second. Succinate have reduction potential of minus 0.03 volts, and final electron acceptor of the complex second is ubiquinone, which is having standard reduction potential of 0.11 volts. So, you can see that in this complex also electron is transferring from lower reduction potential to the higher reduction potential in the gradual way. During this electron transfer, one reduced ubiquinone is produced, but this complex is unable to pump any proton. There are two reasons of it. First is, it is not a transmembrane complex. So, if you want this complex to pump a proton, it must require more delta G, because the proton have to cross to the outer leaflet of membrane. But we have only 0.14 delta I difference, which is not enough to create such a delta G which is required to pump any proton in these circumstances. But the result of this electron transfer leads to the production of one reduced ubiquinone without pumping any proton. That reduced ubiquinone give its electron to the complex third. The interesting point here is, if you see in the complex first, we are producing reduced ubiquinone. In the complex second, we are also producing reduced ubiquinone, but no electron carrier in the complex first having reduction potential higher than reduced ubiquinone, and no electron carrier in the complex second is having reduction potential higher than ubiquinone. Therefore, electron from complex first doesn't pass to the complex third, and electron from complex first doesn't pass to the complex second. So, both produces ubiquinone independently, and that ubiquinone goes to the quinol pool, and from quinol pool it will goes to the complex third. Ubiquinone produced from complex first and complex second goes to the quinol pool, and from quinol pool the ubiquinone will be used by the complex third, where the Q cycle takes place. This has been already discussed in the previous modules, and the Q cycle result into the production of 2 reduced cytochrome C. During this electron transfer, 2 protons are pumped from the complex third, and this proton pump is again a result of electron transfer and production of delta E, which is responsible for the production of delta G. And the 2 cytochrome C that is produced in the complex third, giving its electron to the complex 4, where The final electron acceptor is oxygen, and that is converted into H2O after accepting the 2 electron from cytochrome C. At the same time, the delta E difference that is created in the complex 4 is responsible for pumping 4 proton from matrix to intermembrane space. This shows that electron transfer from lower reduction potential to the higher reduction potential takes place in electron transport chain, and during this electron transport system 10 proton pump from first complex, third complex and fourth complex when we are starting electron transfer from NADH, or 6 proton pumps takes place when we are starting from FADH to our succinate. As the complex first, third and fourth pump proton from matrix to intermembrane space, from intermembrane space to back into the matrix, it is a complex 5 that is responsible for that proton movement. This is known as ATP synthase, which have two subunit FO, F1, and which consist of alpha, beta, gamma, delta, epsilon subunit in F1, while A, B and C subunit in FO. ATP synthase is functionally divided into two subunits. One is rotator, other is stator, and rotator consists of C subunit, gamma subunit and epsilon subunit. Stator consists of A subunit, B subunit, delta subunit, A and B subunit. It is a beta subunit of stator, that is a main catalytic subunit. And other subunit of stator is responsible for supporting the beta subunit, and the rotator subunits are responsible for pumping proton from intermembrane space to back into the matrix. Movement of proton from intermembrane space to back into the matrix leads to the rotation of rotator in one direction. So movement of rotator in one direction causes conformational change in the beta subunit. This conformational change in the beta subunit is responsible for the ATP synthesis. When we are talking about the physics of it, then movement of proton from intermembrane space to the matrix leads to the movement of rotator in one direction that develop torque in the beta subunit, and that torque is responsible for the conformation change. So this conformation change is basically a result of torque developed because of movement of rotator, and the binding change mechanism is given to explain ATP synthesis in the beta subunit. According to binding change mechanism, there are three conformations exist for ATP synthase beta subunit. The first conformation is open conformation, which doesn't have anything. Second is loose conformation, which bind with the ADP plus PI, and third conformation is ATP synthase beta subunit is the ATP binding tight conformation. 3 protons are required to develop a sufficient torque that causes one conformation change in the beta subunit. That means when 9 proton moves from intermembrane space to the matrix, it will leads to the development of 1 ATP in 1 beta subunit. But if you remember, there are 3 identical beta subunits. So the same 9 proton can produce 2 additional ATP from 2 different beta subunits. So we will have movement of 9 proton from intermembrane space to the matrix that will lead to the production of 3 proton per ATP, or 3 ATP per 9 proton. In this way we can say that 3 proton require for 1 ATP synthesis, but we are also requiring 1 additional proton that is moving from intermembrane space to the back into the matrix by a phosphate transporter. So we can conclude here is total 4 protons are required for synthesis of 1 ATP molecule in the ATP synthase. That proton have positive charge. So if the movement of proton take place from matrix to intermembrane space, in the intermembrane space there is an increase in the positive charge. So that will create electrical gradient. So when you are taking electrical gradient and chemical gradient together, they will be responsible for the production of electrochemical gradient. So electrochemical gradient is important and produced because of the movement of proton, but it is very important for the production of delta G for the proton. As you remember that ATP synthesis is not an favorable process. So you must need some delta G for ATP synthesis to complete as a coupled reaction between complex 5 and electron transport system. So the delta G that is created because of electrochemical gradient can be calculated by the equation where delta G is equals to 2.303 RT delta pH plus F delta psi, where delta pH is a chemical between matrix and intermembrane space, or sometime it is calculated when one proton moves from matrix to intermembrane space, and what is the pH difference when one proton present additional to previous proton in the intermembrane space. Delta psi can be calculated in the similar way, but when you are taking together 2.303 RT delta pH is representing the chemical gradient, F delta psi representing the electrical gradient. So when delta pH is known and delta psi is known, then we can calculate what is delta G for the proton in intermembrane space, because R is a gas constant and T is a temperature which is always fixed in a standard condition. At the same time, F is a Faraday constant which is also constant. The delta G which is calculated for the proton in mitochondrial inner membrane was found to be minus 14.2 kilojoule per mole. The important point which you have to remember is, for the production of 1 ATP, we need minus 30.5 kilojoule per mole energy, and you know that we don't have any coupling site here until the delta G of the proton is utilized by ATP synthase to produce ATPs. Therefore, ATP synthesis requires at least 3 protons. If you multiply 14.2 by 3, it is going to be 40.2.6, but you know that coupling is not 100% efficient, so you must have this difference. In this way, if you are calculating what is a delta G that is required for ATP synthesis and what is a delta G that is available with the proton, then you can say that 3 proton is required for synthesis of 1 ATP. And it is also important to note that one additional proton is also utilized by phosphate transporter. So therefore, you can say that 3 proton is utilized for ATP synthesis, one additional is required by phosphate transporter. So 4 proton is required when it transfer from intermembrane space to back into the matrix, then it will synthesizes 1 ATP. Chemiosmotic potential or proton motive force. Peter Michael proposed chemiosmotic theory in 1961. So the movement of proton from matrix to intermembrane space create proton gradient, which is used to generate chemiosmotic potential or proton motive force. So you can define proton motive force as a force that promote movement of proton across membrane downhill to electrochemical potential. Proton motive force help proton to move back from intermembrane space to the matrix. This movement helps movement of rotator in one direction and development of torque in beta subunit of ATP synthase, which produces ATP. And chemiosmotic theory explains the movement of molecules or proton between two osmotically distinct spaces separated by the membrane. So in this case, movement of molecule is the proton and distinct spaces is intermembrane space and mitochondrial matrix, and the membrane is inner membrane. So chemiosmotic theory is actually explaining how ATP synthesis is taking place in mitochondrial inner membrane. Means oxidation of NADH and FADH2, and it seems that when you are saying oxidation of NADH and FADH2 will leads to the phosphorylation of ADP, it seems more better. Oxidative phosphorylation of ADP is dependent on electron carriers. So if NADH is electron carrier, then 10 proton pumps from matrix to intermembrane space, and it will leads to the production of 2.5 ATP, because you know that 1 ATP is synthesized by utilizing 4 protons. Similarly, if FADH2 is electron carrier, then 6 proton pumps from matrix to intermembrane space, and it will produces 1.5 ATPs. So oxidative phosphorylation is also represented by phosphate to oxygen ratio or phosphate to 2 electron ratio. Phosphate to oxygen ratio means how many phosphorylation step you have when one oxygen is reduced into H2O. P by 2 electron ratio means how many phosphorylation step you have when 2 electron transfer to electron transport system in mitochondrial inner membrane. Both are same and synonyms to each other. P by O ratio for NADH as electron donor is 2.5 because we will produce 2.5 ATP, and P by O ratio for FADH2 is 1.5 because we are producing 1.5 ATP. Important thing is we never produce 2.5 ATP, we never produce 1.5 ATP, but it is a calculation, that's why we are writing it. We either produce 2 ATP, we are either produce 3 ATPs. When we are talking about the regulation of electron transport chain, the 2 points come in our mind. The first one is inhibitor of electron transport chain, and the second point is uncoupler of electron transport chain. So first of all, it is important to differentiate between two words, between two molecules. So inhibitors are the molecule that inhibit the ATP synthesis by inhibiting the electron transfer in electron transport system, and uncouplers also inhibit ATP synthesis, but mechanism is totally different. So uncouplers inhibit the coupling that is responsible for ATP synthesis. So coupling where? Between electron transfer and phosphorylation step. So therefore, uncoupler inhibit the ATP synthesis by breaking the interaction between electron transfer and phosphorylation, while inhibitor inhibits the ATP synthesis by blocking the electron transport system. Similarly, we will have different electron transport inhibitor and uncoupler. So first of all, I would like to give you information about inhibitor. So we have 5 complexes. So complex first have inhibitor like amityl and rotenone. If you remember, rotenone is a very interesting, it is used in the fisheries. The second one is malonate. Malonate is an inhibitor of complex second, and then we have antimycin A, that is inhibitor of complex third, and then we have complex 5 inhibitors like azide, carbon monoxide and cyanide, and we have complex 5 inhibitor, that is oligomycin and DCCD. It is important to note that when we are talking about the contribution of a particular complex into the ATP synthesis and you want to study it, then you have to artificially create a liposome and you are going to study in vitro. But at that particular time, these inhibitors are playing very significant role. For example, if you want to see the role of complex first in the ATP synthesis, then you have to block electron transfer from complex third and fourth if your NADH is electron donor or FADH2 is electron donor. So when you want to study complex third, then you have to block complex 4. So therefore, when you are going to study the function of different complexes, then these inhibitors are playing very significant role in terms of study of the electron transport system, but at the same time they are very poisonous. So that is going to be very toxic sometime. For example, if a person is taking azide, it is going to die, and you know the cyanide, they are very poisonous. So they are the inhibitor that is going to be lethal, but they are the inhibitor, they are important to understand the process of electron transfer and ATP synthesis inside the mitochondrial inner membrane. You know that uncoupler uncouples electron transfer and ATP synthesis. Therefore, uncoupler can be synthetic, can be natural. But if you're talking about the properties of uncoupler, it is important to have at least two properties. First one, it should be hydrophobic, that allows uncoupler to transfer in the mitochondrial inner membrane. The second one is it must have ionizable group that allows that uncoupler to take proton from intermembrane space and transfer back into the matrix. So we have two type of uncouplers. The first one is natural uncoupler, the second one is synthetic uncoupler. How uncoupler, it is also important to know how uncoupler works. If you remember, proton transfer takes place from matrix to intermembrane space via different complexes. So in the intermembrane space, that proton is creating some delta G for the proton by creating electrochemical gradient, and when the proton moves from intermembrane space to back into the matrix via ATP synthase, they will produce ATPs. But what happened here, if you have uncoupler, uncoupler takes proton from intermembrane space on its ionizable group and cross the intermembrane space and reach to the matrix via the mitochondrial inner membrane, because they are hydrophobic in nature. So in this way they are dissipating the delta G created in the intermembrane space and leads to the production of heats. So we have synthetic uncoupler, that is 2,4-dinitrophenol, which have hydroxyl group that is going to carry the proton from intermembrane space to back into the matrix. Natural uncoupler are also there. One such uncoupler is thermogenin, another is ionophores. So thermogenin is found in the inner mitochondrial membrane of brown adipose tissues, and they act as a proton transporters. So they transfer proton from intermembrane space to the matrix. The function of the ATP synthase is the same. So they are bypassing the function of ATP synthase, and finally they will allow that proton to be based, because they will not utilize in the synthesis of ATP. Therefore, the delta G associated with that proton is released in the form of heat. And the ionophores are the antibiotics that kills the bacteria, but usually it is produced by one bacteria to kill other bacteria by creating the channels into the other bacteria. And two such ionophores are valinomycin and nigrosin. And if you see the mechanism, they are very interesting. So valinomycin is a uniport of potassium, hence it will disrupt only the electrical gradient without having any effect on the chemical gradient. Because if you add valinomycin in the outer mitochondrial membrane, it will allow potassium to move from intermembrane space to outside the mitochondria. So what happens if a proton moves from matrix to intermembrane space? In the intermembrane space, it creates positive charge, it creates pH difference. But because we are moving one potassium from intermembrane space to outside, that positive charge is dissipated. Similarly, nigrosin is an antipode of potassium and hydrogen. So if you have nigrosin, nigrosin basically transfer one proton from intermembrane space to outside the mitochondria by replacing it with the potassium. So because in the intermembrane space, in the presence of nigrosin, we are removing the proton but replacing with the potassium, so the charges remain same. So electrical gradient is not dissipated, but chemical gradient is dissipated. So in this way, if you know the mechanism of uncoupler, they are very interesting, and they are going to help the study of your production of ATPs by using the synthetic uncoupler. But there are also significance when we are talking about the survival in adverse situations. So student, Let us now summarize what we have learned from this module. So, we have learned what is a mechanism of electron transfer in 4 electron carrier such as NADH dehydrogenase, succinate dehydrogenase, cytochrome BC1 complex and cytochrome oxidase. We have also learned how electron transfer in different complexes are responsible for delta G change that helps proton to pump that helps electron to pump proton from matrix to intermembrane space. At the same time, we have also learned in the intermembrane space how your proton is responsible for development of chemical gradient, electrical gradient. So, if you are taking together, that is electrochemical gradient. We have also learned how this electrochemical gradient is associated with proton motive force, and we have learned how this proton motive force along with the electrochemical gradient together responsible for the ATP synthesis. In the ATP synthesis, by rotating the rotator in one direction, developing torque in the beta subunit and ATP synthesis. So, based on the recent studies, one additional proton that is utilized by phosphate transporters are also considered to be involved in the ATP synthesis. So, therefore, the movement of 4 proton from intermembrane space to back into the matrix is responsible for the 1 ATP synthesis. And you know that when electron transfer take place in different complexes, and we are starting electron transfer from NADH, we are pumping 4 proton from complex first, 2 proton from complex third and 4 proton from complex 4. So, total 10 proton pumps from matrix to intermembrane space, and when they come back into the matrix, they will produce 2.5 ATPs. Similarly, if you are talking about FADH2, so FADH2 can pump 6 proton from matrix to intermembrane space, and that 6 proton is pumped 4 by complex 4 and 2 by complex third. So, total 6 proton pump from matrix to intermembrane space. So, once they come back into the matrix, they will produce 1.5 ATP. So, the number of ATP produced by the ATP synthase in the electron transport system of mitochondrial inner membrane is influenced by presence of inhibitor and uncoupler. So, in this module we have also discussed what is the mechanism of action of inhibitor, how they will alter the ATP synthesis, and how your uncoupler, which is similar to the inhibitor but not the exactly same as the inhibitor, is also going to alter the ATP synthesis. So, in this way we can say that from this module we have learned how a mechanism of electron transport system is responsible for ATP synthesis. At the same time, we have also learned how inhibitor and uncouplers are regulating the electron transport system in mitochondrial inner membrane. Thank you.